each flowering plant, the plant body can be broadly studied by taking into consideration that part of the plant below the ground and that part above the soil level. As you all know, part of the plant which grows below the soil constitutes the root system, whereas that part of the plant visible above the soil is called the shoot system. The shoot system has branches, leaves, flowers, fruits. The anatomy, which deals with the study of the internal structure in the root, in the stem and in the leaves, is variable. Let us now study how the anatomy of the root systems of dicots and monocots are organized, beginning with the dicot root system. The dicot plants have a taproot system. A taproot consists of a main root which branches, forming secondary roots, which further branch into tertiary roots, and there are further branches, whereas in the case of a monocot, the plant exhibits fibrous root system. To study the anatomy of these roots, we have to take thin transverse sections of the roots, stain them properly and examine under the light microscope. When we do so with the dicot root, we see an outermost layer which is the epidermis. In the dicot root, the epidermal cells, otherwise called epiblemma, protrude in the form of fine unicellular outgrowths called root hairs. Below the epidermis, we see a cortex comprising of several layers of thin-walled parenchyma cells. The innermost layer of the cortex is called endodermis. In fact, endodermis comprises of a very thick outer tangential wall made up of a substance called suberin. Suberin is impermeable to water. Besides the outer wall of the epidermis, the radial walls are also filled with suberin, and thus it exhibits stip-like appearance, and these stips are called Casparian stips. Next to the endodermis, in the dicot root, lie a few thin-walled parenchyma cells referred to as pericycle. From the pericycle, initiation of the lateral roots takes place. Initiation of the vascular cambium during secondary growth also takes place from the pericycle. The parenchymatous cells that lie between the xylem and phloem constitute the conjunctiva. All the tissues that are present within the endodermis, such as pericycle, vascular bundles, and the central pith, constitute the stele. Within the stele of the dicot root, we find generally 4 xylem alternating with 4 patches of phloem tissue. The xylem is unique, with the metaxylem facing the pith, protoxylem facing the periphery. That is, the xylem is exarch. Between the xylem and the phloem, there is parenchyma called conjunctiva tissue. As there are 4 xylem alternating with 4 patches of phloem, in most of the dicot roots, the stele is said to be tetrarch stele. Whereas the monocot root in section, just like the dicot root, exhibits similar areas, like it has an outer epiblemma, it has a cortex, it shows endodermis, pericycle, vascular bundles within the pith. However, the roots of dicots and monocots do exhibit differences. As compared to the dicot root, which has few xylem bundles, that is from 2 to maximum 6, that is diarch, triarch, tetrarch, pentarch, hexarch, like that. In the case of a monocot root within the pith, there are more than 6 xylem alternating with the 6 patches of phloem. In most of the monocot roots, the condition is polyarch. The metaxylem are rounded, protoxylem are few in number, as most of the xylem and phloem are at the extreme periphery at the pith. In the center of a monocot root, we see a very large, very well developed pith. Monocot roots do not undergo any kind of secondary growth. As told earlier, secondary growth is the growth in the girth of the plant part, and this secondary growth is mostly the characteristic feature of the dicot plants rather than the monocot plants. Dicot roots undergo increase in their diameter, but monocot roots do not exhibit such a characteristic feature. So thus, though monocot and dicot roots have got almost similar zones when we view them under section, there are differences in the different zones, in the number of xylem and phloem, in the type of the tissue that is present. Let us now study the anatomy of the stem.